This section of the muscle chapter will consider the anatomy of the muscle, how the muscle is arranged as an organ. As with any system, we want to consider how the different tissues that come together make up the organs of our system. And this is especially important talking about muscle because muscle tissue and muscles as organs are different structures. There's quite a bit of muscle tissue in the organ, but there are other tissues also. In thinking about the muscle as an organ, it really kind of comes down to just this one figure as far as how the muscle is organized. The organization of the muscle is sort of a repeating pattern of fiber structures bundled together into increasingly smaller pieces. The entire muscle is a bundle of what are called fascicles, and a fascicle is a bundle of individual muscle fibers, and a muscle fiber is a bundle of individual organelles called myofibrils. Now, these three layers of organization are easily distinguished because they're separated out by layers of connective tissue. The entire muscle is surrounded by a layer of dense, irregular connective tissue called epimysium. A fascicle is surrounded by a layer of dense, irregular connective tissue called paramycm, and then individual muscle fibers are surrounded by a layer of areolar connective tissue called endomysium. Those three words, epimysium, paramycm, and endomysium, obviously all share the root mys in the middle, which is the Greek root that means muscle. And the differences are in the prefixes. Epi means on top of, so this is what's on top of or around the entire muscle. Peri means around, more with the sense of around individual components, in this case, the fascicles that make up the structure of the entire muscle. And then endo means within, in this sense, within the fascicle, around a single muscle fiber. The epimysium and paramycm, like I said, are both dense, irregular connective tissue, which means that they are going to be continuous with the tendon, which is dense, regular connective tissue. And then the tendon is going to be continuous with the periosteum, which is dense, irregular connective tissue, making one continuous sheet of connective tissue connecting the bone to the muscle. The endomysium is areolar connective tissue. It's not part of the structure of tendons, but rather it's just loose connective tissue around the outside of the muscle tissue where blood vessels, nerves, and other structures can get direct access to the surface of the cell. This figure demonstrates the structure of individual muscle fiber cells. They're much like other cells. They contain a number of different organelles, and I just want to highlight some of those. Now, there are some organelles that are exactly like what we see in other cells. They have nuclei. Of course, they have multiple nuclei, but still they're nuclei that contain DNA that can be transcribed to make new proteins, and they contain mitochondria, which make ATP to power cellular processes. A very important cellular process for muscle cells contraction, which requires a lot of energy to be performed. Now, muscle cells have some specialized names for structures that I want to talk about, and then a specialized structure in particular. The cell membrane is referred to as the sarcolemma. The root sarco means flesh in the sense of the flesh of an animal. Now, sarcolemma is really just an alteration of an old name for the cell membrane, which was the plasmalemma. Plasmalemma started being called the plasma membrane, and nowadays it's more often called the cell membrane. But it's really the same thing. The sarco version of it is just what stuck when it was originally named for particular muscle cells. In a similar fashion, the cytoplasm of the cell is called the sarcoplasm. That's not labeled in this picture, but it is true. So all of the space between the mitochondria, the nuclei, and the other structures we'll talk about, all of that cytosolution space, if you will, is called the sarcoplasm. There's another structure that's important for muscle cells that has a specialized name, but it's really just something that all cells have, and that's the Sarcoplasmic Reticulum. Hopefully you can see that that's obviously a modification of the name Endoplasmic Reticulum. Don't be confused. Sarcoplasmic Reticulum is the endoplasmic reticulum, and sarcoplasm is the cytoplasm. So normally in cells, those two words are very easy to distinguish from each other. This is particularly the smooth endoplasmic reticulum, and in muscle cells it stores calcium, which is very important for contraction. The other structure that is specific to muscle cells is called the myofibril. It's really an adaptation section of the cytoskeleton. It contains proteins that are common in the cytoskeleton of other cells, but here they're sort of repurposed to be used to make contractile force occur. So let's look more closely at that structure next. The myofibril is a structure that runs the length of the muscle fiber. It's made up of proteins, many of which we usually associate with the cytoskeleton, but they're arranged in a special way to be able to produce contractile force. The individual unit of a myofibril is what's called a sarcomere, and it's really just the repeating pattern of proteins running the whole length of it. In this picture, we see a bracket around one sarcomere that goes from a structure called a Z-disc to the next Z-disc. Sometimes you'll see the Z-disc referred to as a Z-line. It's a zigzag line, basically, that's connecting all of these muscles together. It's a zigzag line that's connecting all of these proteins together. The two major protein structures within the sarcomere are called the thin filament and the thick filament. We'll look at the structure of those in more detail later on, but thick filaments are made of a protein called myosin, which is related to some of the motor proteins in the cytoskeleton like dynesin and those sorts of things. Then the thin filament is made of a few different proteins. The main one is actin, which is definitely a primary cytoskeleton protein. These thick and thin filaments lay over each other, overlap through parts of the sarcomere. Where the thick filament is found, we call that region the A band. And when you stain muscle cells, muscle tissue, you see the A band as the dark band of the striations. The A in A band is representing the A in the word dark. Then the regions where there is no thick filament is called the I band. That's the thin, light band between the dark bands and the striations. Again, the I in I band corresponds to the I in the word light. Thin filaments extend from the I band into the A band. So the difference in the bands is just where there is or is not myosin. Now, where myosin overlaps with the thin filament, there will be a slightly darker staining quality to that area if it's stained very well. It's not usually seen too clearly in the tissue. But where there's not thin filament overlapping the thick filament, right in the middle is called the H zone, the area that does not actually overlap. Right down the middle of the H zone is what's called the M line, or the middle line. It's what anchors the thick filaments in place at the center of the sarcomere. What we're going to see for contraction is that the thick filaments and thin filaments slide across each other, pulling the two Z-discs in from either side and shortening the sarcomere. This happens along the entire length of the myofibril, and the whole myofibril, and therefore the whole muscle fiber, will shorten. This figure shows a little bit more detail around the myofibril, especially of the Sarcoplasmic Reticulum, and a couple of other features that are important to point out. Now, the Sarcoplasmic Reticulum wraps around the entire myofibril, and every so often it comes to meet up with what's called a T-tubule, which is really just an extension of the cell membrane of the sarcolemma, as represented in this picture. The sarcolemma kind of folds in, and tubes wrap around the myofibrils within the structure of the cell. Where the sarcoplasmic reticulum comes up next to the T-tubule is called the terminal cisternae, which is just an enlarged space storing more calcium. Two terminal cisternae and one T-tubule together make up a structure called a triad. I've said a few times now that the proteins that make up the thick and thin filaments in the sarcomere are adaptations of the cytoskeleton. The first one is myosin, which makes up the thick filament. It's the only one that's in the thick filament. The myosin protein codes for a globular head attached to a long, thin tail. Two of those proteins get wound together, making a double-headed golf club-looking thing. Many of those get laid out together to make up the whole thick filament. At the middle of the thick filament, there'll just be mostly the tails kind of all layered on top of each other. And then the heads will stick out, some of them on the right side and some on the left side, so to speak, of the thick filament. The thin filament is really made up of primarily actin, which again is a cytoskeleton protein, but then also two accompanying proteins called troponin and tropomyosin. Troponin, which in this schematic representation of the thin filament is a single spot is actually a complex of 3 different proteins. One of them is called troponin C. The other 2 are referred to as troponin L and S. I don't remember the C designation for one of them, I remember, but the other 2, I might be mistaken on what it is. The C is memorable because it stands for calcium. That subunit of troponin binds to calcium ions, which will be important later on. The other component of the thin filament is tropomyosin, which is a thin filament type protein that lays on top of the actin, normally blocking a spot where the myosin heads would bind to it. What we'll see later on is that troponin, when it binds to calcium, helps to move tropomyosin out of the way, unblocking that binding site so that myosin can bind to actin and contraction can take place. Consider this question. When you feel that you've reached an answer, hit the next button to go into the next slide, which will reveal the correct answer. The paramycium is the layer of connective tissue that surrounds a fascicle. Epimycium covers the outside of the whole muscle. Endomysium goes around in a single muscle fiber. So in between those is the paramycium.